Hello, I'm Catherine Stevens from the American Chemical Society. Welcome to this news briefing from ACS Spring 2021. We're joined today by Dr. Haibo Huang and Yanghong He from Virginia Tech. They're separating beer waste into proteins for foods and fiber for biofuels. Dr. Huang?
Well, thank you, Kathleen, for this great opportunity for Ellen and I to talk about our research. So our research topic is simultaneous production of 2,3-butanediol and the protein concentrate from blue spent grain. For some of you who are not familiar with blue spent grain, actually it is a waste product or byproduct generated from beer industry. So for most of beer we are drinking, it is made from barley. But the problem is not all components in barley can be fermented to beer. Those unfermentable components in barley, like protein, fiber, lipids, and minerals, they all accumulate and end up in a waste stream called blue spent grain. And currently, blue spent grain is underutilized and is unutilized, and some of them are even sent to the landfills, leading to substantial resource losses and may cause environmental concerns because blue spent grain, they spoils very fast. So we came up with the idea to develop a separation process to separate the protein, fiber, two major components from this waste material. The separated protein could be used as a sustainable and plant-based protein ingredient for animal feed and also to be incorporated into different food products. And we further convert the fiber into a biorenewable chemical called 2,3-butanediol using a novel bacteria called Bacillus licheniformis. And this bacteria was recently isolated by Dr. O'Hare in our research group from the Yellowstone National Park hot springs over there. And this bacteria has a unique property to convert different carbohydrates into 2,3-butanediol at high temperature and high pH conditions. And the 2,3-butanediol is a very valuable compound and has an increasing applications in the pharmaceutical, cosmetics, food, and beverage industries. So in summary, we develop an integrated process to convert a waste material, blue spent grain, from the beer industry to two value-added products, protein concentrate and the biorenewable chemical, to 2,3-butanediol. So we think our approach might increase the sustainability of our society. So in the last, we wanted to thank our sponsors, Virginia Tech Plant and Animal Nutrition Foundation, Virginia Agriculture Council, as well as the USDA SARE Student Research Program. So I'll stop here. Ellen and I will be more than happy to answer any questions you may have.
Thank you both. Are there any questions from viewers? Please state your name and affiliation in the chat window when you ask your question. I'll start with a question of my own. So what gave you the idea to turn beer waste into proteins for food and fibers for biofuels?
Yes, so let me answer this question. When I first started my career at the Department of Food Science at Virginia Tech back to 5 years ago, and I was looking for some interesting project to work with. And our department has a very good relationship with the beer industry. So we occasionally meet with each other and talk about the potential collaborations. And when I was talking to them and what are the potential problems or current issues we could help solve with, so they came up with the blue spent grain. They mentioned that it is they believe this is a very nutritious and resources and material, but somehow it is underutilized. And they have, and if there's no local farmers surrounding, it's a kind of headache for them to ask the waste company to haul them away. So when I looked into the composition of this blue spent grain to see what's inside, why it is currently underutilized, then we found that actually it contains a fair amount of the proteins. It's good. It contains 20 to 30% of proteins inside, but the real problem is it has a very high fiber content, up to 50, 60% of fiber in it. So most of the animals like pigs, chickens, and our humans, we don't have a digestion system that effectively digests such a high fiber content. So that's why we came up with a solution that why not we separate the protein and the fibers out. These proteins could be used for a bunch of applications. Fibers, we could further use it to produce biofuel, because when I did my PhDs and postdoc trainings, I was trained as a fermentation engineer. So we could convert different type of fibers and carbohydrates into biofuels and biochemicals. So this is kind of like to apply or integrate my expertise and with the industrial needs to create such an interesting project. And then Ellen at the time joined my lab, and she is really interested in this project. So we started kind of working together to develop this integrated process.
Great. So another question, how does the alkylase enzyme separate protein from fiber?
Okay, I will answer this question. Because the enzyme can break the band, connected the proteins. So after break the band, the protein become peptides, so they can be easily dissolved in the water. And in the next step, I use a shaking sieve, so the small particle size protein and can be easily separated out.
Is the composition of the protein concentrate similar to other protein products that are plant-based, such as soy or pea proteins?
Yes, in our previous study, I analyzed the amino acids composition of this kind of protein. Also, I compared the amino acids with fish meal protein. So some, especially the essential amino acids, the content is very similar. Yes.
Yeah, so originally the blue spent grain has about 20 to 30% of protein inside and a bunch of the fibers. So things we separate the protein and remove the fibers out, we could really concentrate the protein to up to 45-50% range. That really opens up the windows for the applications of these new proteins. So if we look at the other agriculture materials like the soybean meal, it has a very similar protein content from 40 to 50%. And the soybean meal is currently widely used in the animal feed industry and also incorporate some of the food products. So that's why we think not only separating the proteins, we also concentrate the protein to a high level that allows new applications for this protein from blue or spent grain.
So spent grains are used in cattle feed. If this method that you've come up with were to become widely used, would it create a shortage of cattle feed?
Yeah, that's a great question. We actually, when we were talking about these technologies to different people, and some people did mention that they're concerned that if these technologies became very successful and convert all blue spent grain into the proteins for the other uses, how about the cattle? Because blue spent grain currently is widely used to feed the cattle. So our answer is that we are not trying to convert proteins to the other products. What we do is just to separate the proteins out and concentrate it as a high protein concentrate. So this protein could still be potentially to use to feed the cattle, and also the other components like fibers that has not traditionally be used by other animals, we could convert it to the biofuel. So we are not like taking the food away from cattle. We are more like producing more opportunities for cattle, not only cattle, but also other animals and also human food. And another thing I wanted to say is that for brewers, they don't have the farmers surrounding by, and they have to pay extra money to the waste management company to haul away these waste materials and send it to the landfills. So our technology could really potentially collect this portion of the blue spent grain, which will currently send it to the landfills and to convert it to something valuable. And this protein, collected protein and dried, could be used to send to to feed the cattle and also feed to any other animals and the human people.
So what types of food products have you explored adding the protein to?
I use this kind of protein to replace the fish meal in the shrimp feed. So my study shows that up to 50% of the fish meal in the shrimp feed can be replaced by this spent grain protein.
Yeah, so currently as we mentioned, we have several potential applications used as animal feed and also used as a human food. So we are still in the animal trial stage to look into the feasibility to incorporate our proteins into different animals. And we came up with the first idea to use this protein to feed shrimp, because in the shrimp industry, fish meal is widely used as a protein source. And the fish, if you look at the fish meal's price in the last 20 years, it increased a lot by more than 5 times, all the way to from $200 to $300 per ton to $1,500 per ton. That's created lots of economic burden to the fish feeding or shrimp feeding farmers. So based on our experiment, our protein could replace more than 50% of fish meal in shrimp feed. So we think that could provide a sustainable and a potential low-cost protein source to the agriculture industry. And also we were thinking there might be some other opportunities for using our proteins. For example, we could incorporate the proteins into the protein beverage for human to consume. Or we could incorporate these proteins in different type of food, like cookie, bread, to have the enhanced protein cookies or enhanced protein bread. That's really my dream. We could capture those waste proteins and send it back to the food supply chain. But this, of course, this is kind of our future work, is our dream. It needs lots of the future research, including the product development and the human tasting trials, like sensory evaluations, to see people really like this tasty, like the tasty of the blue spent grain incorporated food.
That's great. You mentioned that your postdoc found a new bacterial species in a Yellowstone Park spring that can convert sugars to 2,3-butadiene. That sounds like a really interesting story. How did that come about?
That was quite a good story. Dr. Josh Ohio, he is from our research group, and he went to the Yellowstone National Park several years ago, and kind of collect a bunch of the bacteria, different species, and we wanted to see that are there any specific unique properties of this bacteria. So we were targeting to separate to use this protein to convert the different carbohydrate to ethanol, another very valuable biofuel product. But we were not quite successful. This bacteria could only produce a very little amount of the ethanol. Then we came up we kind of accidentally found that this bacteria is a very good 2,3-butadiene generator. So we first test it and it works well, and we further test it and works very well. And another very unique property of this bacteria is that it survives very well and works very well at high temperature and high pH conditions. So this will give us a good opportunity for the non-sterile fermentation, because as a fermentation engineer, the most thing we worry about is the contamination, because we want this bacteria to produce certain type of chemicals, but other bacteria also blended in or contaminated in and really mess up the whole fermentation. But this bacteria is very strong at high temperature and high pH conditions, so it could outcompete other bacteria during the fermentation process. This really provides us a very robust fermentation and also the non-sterile fermentation to eliminate the sterilization process, which is very energy intensive.
What about applications for the fiber portion of the spent grain?
The fiber part, like, because it has a high content of hemp cellulose, and it can be used to produce like xylose and xylitol, like this kind of sugar things. And this xylitol also can be used in the food industry. And also because this fiber, it has a high content of carbon, so it can be used to produce active carbon or other cellulose-based nano adsorbent, and which can be used in the wastewater treatment industry.
It's very interesting.
Yeah.
So could this procedure be easily scaled up, and is it cost effective?
Yeah, so this when we first started our project, we really use a very small scale, like use beakers and shaking flasks, dealing with a very tiny amount of the blue spent grain, like 5 to 10 grams. So after a few years of the verification of the concept and approval of concept, now we kind of scaled up this process by about 100 times. So which allows us to produce 5 to 10 pounds of the proteins and the fibers from the blue spent grain. And this protein and fibers, as we could allow us to do the further testing, like the animal feeding testings, and also for the fermentation testings to convert fibers to 2,3-butanediol. But so the beautiful part of this process is very simple, scalable, but very effective to separate the protein and the fibers out. But we also realize that 5 to 10 pounds means a lot to us, but it still means very small scale in terms of future commercializations. So in the